And these human stem cell derived cardiomyocytes, they enable the testing of these therapeutic strategies in the molecular context of human cells. And then our preliminary data shows partial rescue of the calcium handling, and we want to test new design strategies that might enable larger variants to be expressed. And with that, I'd like to thank the people who contributed to these studies and my funding, and I'm happy to answer any questions.
Wow, that was super interesting. Thank you so much. So I have the microphone, so if anybody has questions, I can bring the microphone to you so that our speaker can hear your question. So questions. All right, while you think about your questions, I'm actually going to start with one. So I think that's so interesting to think about how everybody's trying to use viruses to encapsulate the gene that you want to deliver and then get that into the cells. And I was wondering, it's like you kind of mentioned people have focused on AAV as a particular kind of virus that they've used for a long time. But as we get further along with, protein engineering, viral engineering, those kind of things, are people going to try to create like an engineered virus that would be able to have a larger DNA, payload, as you mentioned, so you could get a bigger gene, like the whole dystrophin gene in a virus? Or are there just limitations to what the cell will take up or something like that that make it hard to do that?
So there are definitely people in that space, just evolution of viruses, and then also, so for example, Myosana is a company that is focusing on lipid nanoparticles and trying to basically decorate that nanoparticle with ligands that will be taken up by the muscle. And then the idea is that, eventually, right now, as the technology stands, it's abysmal compared to AAV, but those approaches are definitely being explored.
Okay, questions from the audience?
Thank you. Hi, I was just wondering whether you thought that in addressing the cost issue of the production, you think it would be more viable to target the manufacturing side or just finding a more effective solution as a whole?
Yeah, so the manufacturing process is, kind of a boring question, but I'm certain that different pharmaceutical companies are in that space. One issue is the ultracentrifugation, and but there's also just, you have to produce virus using cells. And then so being able to transfact the cells with the different components that make the packaging process successful, that's also a space that's also being explored by, I wouldn't say exclusively by industry, but industry is more motivated to bring those costs down.
Hi, Dr. Gucci. It was a very interesting talk. I actually learned a lot. So I guess one of the questions that that emerged as you were giving the talk was, I was wondering whether or not there are any known functional, like qualitative differences in the protein product of the microdystrophins and how they compare to the regular length dystrophin.
You're talking about the protein function.
Yeah.
We know that it localizes to the right place. It goes to the sarcolemma where it's supposed to go. And there's very little information on the structure of the protein, just because it's crystal structures are really hard to obtain for dystrophin. There's been parts that have been crystallized, but not the whole thing. And so in terms of function, there are some papers that address specific domains that might be better, and it's still up for debate whether recruiting nitric oxide is actually helpful. There's an argument that it's important for the heart, but that evidence seems to be very weak when I surveyed the literature. So it's being marketed that way, but yeah, there's still a lot to be explored. There's one paper that discusses how hinge 2, which is included in the Sarepta product, actually causes problems in the attachment of the nerve to the neuromuscular junction and the myotendinous junction. And then when you swap hinge 2 with hinge 3, then you start to, you basically, all of those problems go away. And so there are certain domains where we know more information, but not every domain has been systematically swapped out like that.
Thank you.
I really enjoyed your talk. Thank you. My question's sort of in the same vein as Dr. Flanagan's question. Are there other candidate viruses that you could potentially use for this, or are they too difficult to work with?
So the beauty of AAV is it's not integrating, so it stays episomal, and so that is what the FDA wants. They don't want non-integrating, because if you have integration that is random and it could potentially drive an oncogene, then that would cause problems. Although, there is an argument for assessing whether a patient is at risk for cancer, but especially because these patients don't live very long. And in terms of different viruses, I mean, all of them have been explored, and then AAV is right now the most efficient way to systemically deliver. So if you're talking about intravenous injection, this is the way to go.
Thank you so much.
Okay, I have one more question. So dystrophin is such an interesting protein because it's so large and it has all these different domains that do these different functions. And so, you mentioned earlier that you're trying to make these smaller ones and hope that the proteins link up with each other. And I was wondering, how far do you think we could push that? Do you think it would be possible eventually to have something where you put in, 5 different pieces or, 10 different pieces, and they have particular sequences at their edges that tell this one to link up to this one, and then the next one to go onto the chain until you can build the whole thing?
Yeah, so it is definitely possible to get 3 different ones to link up together, and but the challenge is getting 3 different components in a cell. And so patients are already getting the borderline toxic dose, and just to get one. And then, so in order to increase the probability of getting 2 different ones to get into one cell, you would have to dose the patient with more. And so right now, that's not possible. But maybe with these more myotropic vectors, we can lower that dose required.
That makes sense. Okay. Other questions? Okay, let's thank Oscar again.